we're going to look up some protein sequences using this tool from the government. Thank you, government. So this is dystrophin, which is the gene for the protein that, when mutated, causes muscular dystrophy. You can see it's in all different types of organisms. So I'm just going to type in dystrophin Homo to get Homo sapiens. If I look at this gene and I want to know the sequence, I can click to get the sequence. I haven't used this website in years. Here it is. So this is the DMD. This is a gene that's on the X chromosome. That's why it's an X-linked disorder. So only boys get muscular dystrophy or Duchenne muscular dystrophy because girls have two copies of the gene. They have an X copy and a Y copy, or an X copy and an X copy. Boys only have an X copy, one copy, because they have a Y chromosome. So if you mutate the one copy, there's no backup copy. Females have a backup copy. So here's the gene sequence, and you can see it's a very intricate detail, but one, and it goes on and on. In fact, dystrophin is one of the biggest genes in the human body. One misspelling of this, just one of these, right? How many are there? What does it look like? Look at the bottom of my screen. You can see the scrolling is going up very slowly. What would you guess, 1 million? So there's all kinds of tools in here to go through it, but it's an enormous protein. And there's all kinds of different ways you can look at these. And again, if you want to do this yourself, just go to the NCBI. But yeah, I think that you can get a sense for just, this is a very large protein, or gene, I should say, but there are many genes that are smaller. There's most genes, in fact, are a lot smaller than this one. But you can see that this gene that it codes for this important protein called dystrophin, it's such an important protein when it comes to muscle that a single wrong nucleotide can cause death, unfortunately, in people. So let's keep going. All right, so amino acids are linked in different sequences, giving each type of protein molecule a different three-dimensional shape or conformation, just as different sequences of letters spell different words. In this way, the same basic biochemical machinery has served to generate the whole gamut of life on Earth. Right, in all living cells, genetic information flows from DNA to RNA and from RNA to protein. That's transcription from DNA to RNA and translation from RNA to protein, an arrangement known as the central dogma. The sequence of nucleotides in a particular segment of DNA, which is a gene, is transcribed into an RNA molecule, which can then be translated into the linear sequence of amino acids of a protein. Only a small part of the gene, the RNA and protein, is shown. All right, living cells are self-replicating collections of catalysts. That's a great way to put it. One of the most commonly cited properties of living things is their ability to reproduce. For cells, the process involves duplicating their genetic material and other components and then dividing into two, producing a pair of daughter cells that are themselves capable of undergoing the same cycle of replication, sort of an exponential growth. It is the special relationship between DNA, RNA, and proteins, as outlined in the central dogma, that make this self-replication possible. DNA encodes information that ultimately directs the assembly of proteins. The sequences of nucleotides in a molecule of DNA dictates the sequence of amino acids in a protein. All right, proteins in turn catalyze the replication of DNA and the transcription of RNA, and they participate in the translation of RNA into proteins. This feedback loop between proteins and polynucleotides underlies the self-reproducing behavior of living things. So this is interesting, and there's one author named Douglas Hofstadter, I think, who wrote a book called 𝗙𝗪𝗓, which is a very influential book. I recommend the book. It's good, not great. And anyway, the point that he makes is, and many people have observed this, is that when you look at, hold on, my cat messed up a lot of my infrastructure here. When you look at DNA, it's self-referential. What does that mean? This central dogma is called dogma for a reason. Dogma is usually used like kind of an insulting word, right? Well, the reason this is kind of interesting is that this is the central dogma, but the reality is a bit different. There's there's a lot of other interactions that are occurring. I won't talk too much about those just yet. That's what this whole book is about. But what I want you to understand is that, sorry, I don't know what my computer's doing right now. What I want you to understand is basically this idea that when some of these proteins, right, are related to helping DNA replicate and maintaining their status. So there's this feedback loop that's very interesting, right? There are proteins that help DNA replicate. And those proteins are created by the very DNA. So some people call this recursive in computer science. There's a lot of concepts like this in art and music, which is why it's called 𝗙𝗏 And Gödel's incompleteness theorem has a recursive idea in it. So and Hofstadter's general theme of the book is that this is where like all complexity and intelligence and beauty lies in this self-referencing sort of strange loop is what he calls them. He even has something called I Am a Strange Loop. I think it's a book, and maybe even became a musical, I don't know. But anyway, it's an interesting book. But the point is, you really can't have DNA without the proteins that allow the DNA to have its elegant song and dance of this is some DNA, of basically chaperoning it to divide and separate and then rewind again and come apart and then having the cells kind of divide. This is two cells dividing. It starts off as one cell, and you can see them kind of budding out, and then they separate. Proteins are required to do this, but wait a second, the data for the proteins are stored in the DNA. The RNA is kind of like this intermediate. So, it's sort of like this very funny thing. Well, if DNA comes from proteins and proteins come from DNA, how does this all work? And it's kind of amazing to think about. And I think the thing about Hofstadter, though, is a good scientist and everything, but, the fact that this, like, blew his mind is probably a little, like, memey, and it kind of reminds me of, like, maybe the stereotypical pothead or something. Saying, whoa, bro, this is crazy. The reality is it's very explainable and not that deep as maybe the connections that he's drawing, that he thinks they are. So anyway, that's just my 2 cents. So let's keep going here. DNA encodes information that ultimately directs the assembly of proteins. The sequences of nucleotides in a molecule of DNA dictates the sequence of amino acids in a protein. Proteins, in turn, catalyze the replication of DNA and the transcription of RNA, and they participate in the translation of RNA to proteins. This feedback loop between proteins and polynucleotides underlies the self-reproducing behavior of living things. We discuss in complex interdependence between these 3 actors in detail in chapters 5 through 8. In addition to their roles in polynucleotide and protein synthesis, remember, polynucleotide just means DNA or RNA, a lot of nucleotides. Proteins also catalyze the many other chemical reactions that keep the self-replicating system shown in 1.4 running. So let's see what 1.3 is here. These are all living organisms are constructed from cells. This is a colony of bacteria, again, 2 micrometers, not quite visible, and bacteria could be good or bad. A butterfly, a rose, and a dolphin are all made of cells that have a fundamentally similar chemistry, which is really amazing that the DNA, if you compare the DNA amongst these 4 organisms, they're very, 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 very similar. Which surprised me when I learned that the DNA contents of a butterfly and a dolphin and a rose, you can imagine these things are so different. Most of the machinery is the same. Most of the way that we harvest energy and things like that has not changed in a billion years, which is really kind of amazing if you think about it. Or maybe 500 million years, better way to look at it, or 100 million years. All right. A living cell can break down nutrients and use the products to make both the building blocks needed to produce polynucleotides, DNA and RNA, proteins, and other cell constituents. What's in a cell? We'll find out soon. And to generate the energy needed to power these biosynthetic processes. So it's using biology to make things. We discussed these vital metabolic reactions in detail in chapter 3 and 13. This word metabolic confuses a lot of people, but basically metabolic is just any kind of biosynthetic process. And we sometimes say metabolism. Some people say, well, I have a fast metabolism or slow metabolism. And I think that's, to me, is often in their head. But the point is, any transformational reaction from one entity to another can be thought of as a metabolic reaction. Let's look at figure 1.4. Life is an autocatalytic process. So auto means automatic or will occur on its own. Catalytic means something happening or something being created or transformed. DNA and RNA provide the sequence information, the green arrows, that is used to produce proteins and to copy themselves. Proteins, in turn, provide the catalytic activity needed to synthesize DNA, RNA, and themselves. So it's amazing that the proteins are kind of machines that make all this happen, and the DNA is the information. It's almost like the software and hardware to some extent. Together, these feedback loops create the self-replicating system that endows living cells with their ability to reproduce. And as you study this, I think you'll find it to be rather amazing how you can have a protein that's sort of guiding, or even dozens of proteins or hundreds of proteins that are guiding and shaping the DNA and RNA so that the DNA and RNA can transform into more protein. So the very proteins that are guiding the DNA and RNA are sequenced from DNA and RNA. So there's no top level, if you will. It all starts sort of at DNA, I suppose, but there's still kind of this very intricate meta transformation. You don't usually see that in things like computer programming, where you sort of have a core level of DNA, and typically, I suppose well, maybe it is somewhat similar. I don't know. It's something very interesting to ponder and think about. Only living cells can perform these astonishing feats of self-replication. Viruses also contain information in the form of DNA and RNA, but they do not have the ability to reproduce by themselves. Instead, they parasitize the reproductive machinery, or they take over the reproductive machinery of cells that they invade to make copies of themselves. This is like the COVID virus will take over some of your cells to make a copy of itself. Thus, viruses are not truly considered living. They're merely chemical zombies, inert and inactive outside of their host cells, but able to exert a malign or malignant control once they gain entry. We review viruses in chapter 9. Yeah, if you look at my DNA, there's loyalty instead of A, C, T, and G, but that's another topic. Scientists are very confused by that. All living cells have apparently evolved from the same ancestral cell. This is a very, very unusual idea. All living cells have evolved from the same ancestral cell. When a cell replicates its DNA in preparation for cell division, the copying is not always perfect. I think this is probably thing number 2 to really understand in biology. This will help you understand a lot. When a cell replicates its DNA in preparation for cell division, the copying is not always perfect. Well, why? Well, we just looked at the NCBI, right? NCBI, National Center for Biotechnology Information. We can go to the genome domain or gene domain. We can type in, I don't know, insulin. Do you want to type in insulin? Any other protein some people care about? There's insulin, there's the insulin receptor, there's the insulin precursor. I'm going to type in insulin Homo, again for the species, but I could do Mus musculus, which is just a mouse, and we could see the insulin gene in mice. We can look at KRAS, which is a very unusual protein. Insulin is most definitely a protein. It's made from an insulin precursor. So let's look at, and then it has a receptor, and there's IGF, which is not insulin, but related. Here's an insulin degrading protein. You can see there's dozens of proteins related to insulin, but the big one is called pre-pro-insulin, I think, or pre-insulin. I think it might be. But a lot of hormones have a pre-pro kind of concept within different, let's see. There's pre-insulin, pro-insulin. There's pre-pro in the elephant shark. Well, this is plain old insulin on chromosome 10. So maybe insulin doesn't get chopped up and metabolized. Anyway, let's go to the genome sequence. Here's some of the ways to look at the sequence, but I'm just going to go to the basic FASTA, which is just the letters, because I think that's better. There it is. It's really small relative to dystrophin, right? Dystrophin was like millions of base pairs. These are each called base pairs. And you can see there's 4 letters, A, C, T, and G. So anyway, insulin is really, really short. And we're going to learn in different chapters that these three nucleotides each code for a different amino acid. And this is around 44 amino acids, if I'm not mistaken. So we're going to determine and learn a bit about this down the road. Why did I mention this? Because when we looked at dystrophin, which was so large, it made sense to me, at least, that it was possible that you couldn't necessarily replicate this too easily, right? That maybe something would go wrong. Millions of base pairs. I can't even scroll down fast enough to get to the bottom. This is maybe the fastest I can scroll, and it would still take me about a minute to scroll down to the bottom. So how could it be possible that this gets copied perfectly every single time? It would have to be a really good copy machine, right? We have this copy machine inside of our body that is this good. And when we think about computers, we think about copying and pasting. When you copy and paste something, it's not like one thing in the copy paste will go wrong, right? It's not like one letter will accidentally get changed. Well, that's because our computer software is a little bit different from our body software. In our body software, every now and then, one of these will, instead of an A, T, C, or G, it might go to an A, C, T, or G in a different order. And that's all it takes for there to be a so-called mutation. And you can see just how long this was, right? This is so enormous. How do you get it exactly the same every time? That's sometimes called fidelity. And we use that term in music or audio sometimes, hi-fi, lo-fi. So high fidelity is a perfect copy. Well, on occasion, the instructions are corrupted by mutations that change the sequences of nucleotides in the DNA. For this reason, daughter cells are not necessarily exact replicas of their parents. Mutations can create offspring that are changed for the worse, in that they are less able to survive or reproduce, or changed for the better, in that they will be better able to survive or reproduce, or changed in a neutral way, in that they are genetically different but equally viable. The struggle for survival eliminates the first, favors the second, and tolerates the third. The genes of the next generation will be the genes of the survivors. That's probably the next most important thing to understand in biology. Central dogma: cell replication does not result in a perfect copy, and the genes that survive are the genes of the survivors. Now, we notice that there are still genetic diseases despite this, right? And that's because the window for reproduction and survival are two different things. You can have Duchenne Muscular Dystrophy, which has a lifespan of around 20 years, and still reproduce. You can have a lifespan of 40 years with Huntington's disease and still reproduce. So it's not a perfect sort of one-to-one ratio. If you have a fatal disease that results in death before the age of 5, then the genes of the next generation will be the genes of the survivors and not the genes of that patient, for instance. So anyway, I'd encourage you to think about this a little bit, that it seems to be that most mutations are actually ones that change you for the worse. Having said that, the only ones that really carry on over long periods of time are the ones that change you for the better. So it's a very interesting sort of concept. For many organisms, the pattern of heredity may be complicated by sexual reproduction, in which two cells of the same species fuse, pooling their DNA. The genetic cards are then shuffled, redealt, and distributed in new combinations to the next generation, to be tested again for their ability to promote survival and reproduction. One way to look about or think about DNA is basically DNA is just a game. And the game is, can you reproduce and send your DNA? Here's some DNA here. Here's some DNA here. And the game is, can this DNA reproduce better or more than this DNA? And that's all there is to life, is who can reproduce the most. And if you think about it that way, it's a little bit of a cold world, but it is definitely the kind of dynamic that is sort of what gives rise to life. This DNA is competing for resources. The machines that it makes, the DNA makes proteins. So those proteins are molecular machines. It's trying to make the best molecular machines that will reproduce itself. If it can't do that, it'll die. So this DNA might make a protein organism of a cat. Well, this one made a cat, but a cat with no claws. Well, this one's going to survive and reproduce, more likely at least, than this one. And maybe that's why cats have very sharp claws. And so forth and so forth. And all the way up to perhaps even consciousness. And there's a lot of debate as to whether consciousness, what we seem to exhibit, is a byproduct of evolution or direct cause of evolution. And we don't know. But it's a good question.